Welcome, everyone. PPMD is happy to host a webinar to discuss the top-line results from Sarepta's phase 3 EMBARK study. As you'll recall, this is the gene therapy of Sarepta, and there is a current approval of this product for 4 and 5-year-olds. So we're really happy to have Sarepta join us and Dr. Crystal Proud. Next slide, please. So with us today are Dr. Crystal Proud. I'm not going to read her bio, and it's actually much longer than this, but I think many of you probably online know Dr. Proud from Children's Hospital of King's Daughters in Virginia, and we're happy to have her join us along with Siobhan Fitzgerald, Executive Director of Patient Affairs for Sarepta Therapeutics. And I think you've probably met both of these lovely people at some point in time, whether it's in the clinic or in meetings or certainly online. So with that, I welcome Dr. Crystal Proud, and she's going to begin with the top-line results. Thanks, Crystal.
Certainly. So I really appreciate the opportunity to share some exciting data today and provide some clinical insight into what I see as really a transformative therapeutic landscape for Duchenne muscular dystrophy. Here are my disclosures. I do serve as a primary investigator for several of the clinical trials involving the product that we'll be talking about today. We'll be discussing gene transfer therapy as it relates to recent data from the SRP 9001-301 clinical trial, or EMBARK. And so let's start first with just reminding ourselves about the mechanism of action of gene transfer therapy. Remember that this is whereby a piece of DNA has been packaged inside of a viral vector or a viral capsid in order to be delivered to the patient for treatment. So this viral vector is administered via, in this case, an intravenous infusion on one occasion, and it carries the transgene or this piece of DNA into the cell where we desire it. That transgene material is then released into the nucleus, where something called transcription happens, and it turns that DNA into a piece of RNA. That RNA floats out of the nucleus, goes into the cytosol, and then leads to protein production. And typically we're looking for this protein to be the protein that the patient's body does not make naturally on its own, perhaps because of a mutation in a gene. In this case, we're going to be talking about dystrophin that the patient's body is not making on its own. There's a variety of different outcomes that are utilized when we're looking at gene transfer therapy in Duchenne muscular dystrophy. So safety of course, is first and foremost a priority. And then we can look at functional responses. We'll touch on several of these today. And then there are measures of protein expression as well as transduction. But today we're really going to focus on these top two outcomes. In order to provide some framework around these functional outcome measures, let's remind ourselves of the North Star Ambulatory Assessment in Duchenne muscular dystrophy. So there has been some really fantastic natural history studies that have demonstrated to us the change over time in the North Star Ambulatory Assessment score for our boys affected by DMD. Usually we see some improvements in the score whereby it reaches a peak on average of 26 at age 6.3 years old. And then we traditionally would expect to see a decline in that score over time. On the right-hand side of the slide here, you can see the components of the NSAA. And so they're in order of the least difficult to the most difficult. And then they are given a score of 0 to 2. Now, this means that this score is very concrete. A score of 0 means that the boy is unable to accomplish this task at all. A score of 1 means that he is able to accomplish the task, but requires some sort of modification and does not do it in a completely typical fashion. And a score of 2 means that he does this maneuver in a completely typical fashion. And we'll come back to why this is a helpful tool, but also why it may not be the perfect tool for us to be able to provide insight into certain measures that we're looking at in Duchenne. But this gives us a framework because this is going to be utilized as our primary endpoint in this study. As was mentioned, delandistrogene moxeparvovec is the only currently FDA-approved medication, which is a gene transfer therapy for Duchenne muscular dystrophy, and it's applicable currently for boys ages 4 and 5 years old. Whenever we're prescribing a medication, it's important for us to keep in mind safety considerations. This medication is contraindicated in patients with a deletion in exon 8 or exon 9. In the clinical trial programs, acute serious liver injury has been observed. And so as a result, it's important for us to pursue laboratory measurements at baseline and then following infusion of this medication. And we also utilize a corticosteroid regimen throughout the treatment period, as specified in the prescribing information. In clinical trials, immune-mediated myositis has been observed, particularly for patients who had involvement of mutations with exon 8 and exon 9. And so this is why it is currently excluded. And so this is something that we monitor clinically for. And then acute serious myocarditis and troponin I elevations have also been observed following administration of this therapy. And so as a result, it's important that we monitor our boys clinically after treatment, and then also with laboratory monitoring, once again, as specified in the prescribing information. Because this is an AAV vector-based gene therapy, it's important for us to make sure that when we're treating a child with gene transfer therapy, that they do not have pre-existing antibodies that would impact the efficacy of that therapy. And so you all well know that we pursue antibody testing prior to consideration for treatment to make sure that a boy is eligible for this therapy. Now, the most common adverse reactions that have been observed with delandistrogene moxeparvovec are those you see listed here, which includes vomiting, nausea, liver function test abnormalities, fever, and reduced platelet count. So let's get into the EMBARK study design at this point. So we see here that this was a phase 3. It is ongoing. It is a multinational, double-blind, randomized, and placebo-controlled study looking at safety and efficacy of this gene transfer therapy compared to placebo in boys with Duchenne ages 4 to 7. And so you can see the construct here. These boys were randomized after enrollment, and they had to have an NSAA that was between 17 to 28. They had to have a time to rise from the floor. So in my clinic, I call this a supine to stand time of less than 5 seconds, which is quite a rigorous baseline to have. We've mentioned that they had to be between 4 and 7 years old, and they were stratified into different groups based on these age groups, as well as based on their baseline North Star Ambulatory Assessment score. The way that this clinical trial worked was that in the beginning of the trial, in part 1, which is what we'll discuss today, a certain number of these boys were placed into the group randomly that would receive gene transfer therapy first. So this would be 63 boys in this case. And then 62 boys were randomized to receive an IV infusion of placebo. And they were followed over this first 52-week time period. And at the end of that 52-week time period, they would flip-flop, whereby those boys that received placebo would move into the group that would receive delandistrogene moxeparvovec. And then likewise, the boys that received gene transfer therapy in part 1 will receive placebo in part 2. The primary endpoint, which is a specified assessment before the trial even begins, was identified as a change in the North Star assessment score from baseline to week 52. So we'll talk about that. And then there were some key secondary endpoints and some other assessments that were pursued throughout the clinical trial program as well. Now, when designing a clinical trial whereby one group that receives a treatment is compared to a group that does not receive treatment or receives placebo, the goal is to have a well-matched group at baseline so that they can be accurately compared. We can see here that baseline characteristics, including items such as dosing weight and time since diagnosis of Duchenne, were not significantly different between the two groups at their baseline. In addition, there was no significant difference in the timed function tests at baseline between the two groups either. And this means that the two groups were well matched at the start of the study. So let's discuss the results of part 1 of this study, whereby we're comparing the group that received delandistrogene moxeparvovec to the group that did not receive gene therapy. And we're looking at how they did over this first 52-week time period. When looking at this plot, it's important to know that the vertical line in the middle would indicate that there is no difference between treatment or placebo. Results to the right of the midline would favor an outcome that is attributable to treatment with delandistrogene moxeparvovec, and results to the left of the midline would favor placebo. The dot here indicates the mean or the average. And if you look at the chart to the right, you see a column that is labeled p-value. And in general, we consider a p-value of less than 0.5 as being statistically significant. So let's look at the primary outcome, which was the change in North Star Ambulatory Assessment Score at 52 weeks. What's clear is that while the p-value to the right is not less than 0.5, the change in North Star favored the treatment group. And if we shift now and we look at the key secondary endpoints, our timed functional testing like time to rise, 10-meter walk run, stride velocity, and ascending 4 steps, you can see that these also favor the treatment group with delandistrogene moxeparvovec. And so this is impactful for us to consider. These functional benefits of delandistrogene moxeparvovec are not limited to a particular age group. And so the p-values for both the 4 to 5-year-olds and the 6 to 7-year-olds in these key secondary endpoints demonstrate statistical significance. And let's dive a little deeper into some of this key secondary data here. We'll start with the time to rise from the floor. So many of you have seen this conducted in our clinics. This is where we have a boy lie all the way on the ground, and we time them to see how long it takes for them to assume a completely upright position with their hands to their side and their feet together. Treatment with delandistrogene moxeparvovec is in purple, and placebo is in yellow. Improvements in the time to rise from baseline, which would be a faster time, are indicated above the midline in this representation, and a longer time to rise would be indicated by a drop below this line. So the group overall, which includes the 4 to 7-year-olds, shows improvement in the treatment group with a faster time to rise and a longer time to rise from the floor in the group that did not receive treatment. So this difference was statistically significant, that p-value being less than 0.5 when we compare those two. If we separate out the two age subgroups, we see that the 4 to 5-year-old group that received treatment improved in their faster time to rise, while the untreated group was essentially stable. Not unexpected for this age group, this 4 to 5-year-old age group. And the difference between these two groups was also statistically significant. Let's look at the panel all the way on the right. The 6 to 7-year-old group that received treatment showed stability, while the untreated group showed a decline with a longer time to rise from the floor, which I might expect just given the natural history of what we know occurs in this age group. And the difference between these two groups was also statistically significant. Similarly, the 10-meter walk test showed faster times in the treatment group compared to no notable change in the untreated group in this overall 4 to 7-year-old group at 52 weeks. And remember that this is a test whereby we've marked out 10 meters and we're asking a boy to walk or run if he has the ability from the starting point to the end of that 10-meter point, and we're timing him. The difference between these two groups was statistically significant in that overall age group. The 4 to 5-year-old subgroup showed improvements in the time to walk 10 meters over 52 weeks for both the treatment and the placebo group, but the treatment group here did better, and so much better that this was also statistically significant. In the 6 to 7-year-old subgroup, the treatment group showed stability in the time to walk 10 meters, while the untreated group took longer to walk 10 meters. And the difference between these two groups was statistically significant. So if we're seeing these statistically significant differences in key secondary endpoints, how does that relate to the primary endpoint? And so let's go back to our discussion about the North Star Ambulatory Assessment Score. When a boy cannot accomplish a task, they get a 0. And when they accomplish a task in a very typical fashion, as a boy would who does not have muscular dystrophy, they get a 2. So that 1 range really has a lot of variability in it. And so improvements in the quality of movement or the speed of movement may not be adequately captured by this North Star, because we may see that they are faster, or in my case, in some of my boys that were treated in one of our open label clinical trials with delendistrogene, I noticed that very shortly after treatment, I could see a difference in their quality of movement when they were rising from the floor. So they stopped turning over 180 degrees, assuming a wider base in their posture to be able to get upright. And so they would still be given a 1, even if the quality of their movement improved. And so while the North Star is a fantastic tool for us to use overall, it is these subtleties that make it imperfect, just like many of our tools in medicine are. And so we have to keep that in mind when we're assessing these outcomes. So once again, safety is priority as we pursue any treatment. And what's important to know here is that the adverse events seen in this clinical trial were consistent with the safety profile from previous clinical trial experience, including those safety considerations that we've already reviewed at the beginning of our talk today. Importantly, there were no complement-mediated adverse events, there were no deaths, and no discontinuations from the program. So how do I interpret these results when considering the boys that I take care of in my neuromuscular clinic? Let's take a look at the secondary endpoint of time to rise. This was intentionally chosen as a secondary endpoint, as the first key secondary functional endpoint, given that it is the most responsive marker of disease progression in earlier stages of Duchenne muscular dystrophy. A 5-second rise time is a threshold for predicting disease progression, indicating a functional decline. And so it's a strong prognostic indicator. It helps to guide our discussions with our families in clinic. And here we see that the likelihood of losing the ability to walk is increased with a rise time of more than 5 seconds, whereby that risk reduces as rise time is quicker. So the faster you can get up off of the floor, especially if it's less than 5 seconds, that is significant for us to be able to have some prognostic information about how long our boys will continue to walk. So treatment with delendistrogene reduced the odds of progressing to a rise time of greater than 5 seconds by 91% in the overall group. And if we want to look at this older group, perhaps the more risky group, we see that at week 52, there were only 6% of boys with a rise time of more than 5 seconds in the treatment group, compared to 25% of boys in the untreated group or the placebo group. And so this means that in a 6 to 7-year-old age group, the reduction of risk with treatment was 89% lower likelihood that these boys would progress to a time of more than 5 seconds. And based on our natural history knowledge, this is impactful when we consider the potential for this to change the time to loss of ambulation. So in summary, while the primary endpoint was not met in this initial 52-week data cut from EMBARK, the data here demonstrates substantial evidence of effectiveness of delendistrogene moxeparvovec, including impact on very key secondary endpoints that are known to be predictors of disease progression. And in addition, the safety profile remains consistent with what we've seen throughout the clinical trial program. So with that, let's begin some questions and some discussion.
Crystal, thank you. And I know Siobhan is going to be joining us. And first of all, thank you. That was a very terrific and concise and clear representation of the EMBARK top-line data. So I think maybe just because we're all here, let's just have a conversation, and I'll toss questions between you and Siobhan if that's all right. And sometimes I'll refer back to slides, but in general, I think we can have just a general conversation. For those of you online, I know you have lots of questions, but we've received many, many questions prior to this webinar. And so I'm going to try to get to as many of them as I can during this hour. So first of all and foremost, the current label reads as for treatment of 4 and 5-year-olds, as you know, and there are exclusions of exons 8 and 9. So Crystal, I'm going to direct this to you and then to Siobhan. The question is, we're still excluding them by label of those treated under Elevidys, under the name it's been given following approval. Do you see any other exons that worry you in this, which is a question that has come in? And then to you, Siobhan, about how it will Sarepta consider, if this remains in the label, how will Sarepta be considering people with mutations in that area? So first you, Crystal.
That's an excellent question. And I think that it's been incredible to see the collaborative work that's been done from companies in this space working together to try to identify and mitigate risk. And identifying these exons of most risk has been part of that collaborative process. And so at this point, it does appear that excluding mutations, deletions in particular, in exons 8 and 9 is the safest way to proceed in this moment, because it would indicate a higher risk for the potential for immune-mediated myositis. Now, based on the clinical trial experience, there are some areas that we have much more experience in as far as with regard to specific exon experience. And there are some areas that have less experience, 1 to 17, for example. And so it doesn't mean that there are other exons that are completely without risk if your child has a mutation in that area. This exon 8 and 9 exclusion is purely because of the identified risk for immune-mediated myositis in particular. But as with any new approved therapeutic, it's important for us to be able to monitor safety and to keep an open mind, observe clinically, observe from a laboratory perspective, to make sure that we have our eyes open, and if we see an adverse effect, we can address it.
Thank you, Crystal. And so you mentioned this immune-mediated myositis. Can you shed a little light on, those are pretty big medical terms, and if you can just explain that, that would be really helpful.
Absolutely. So the way that I think is easiest to think about immune-mediated myositis in this case is, remember that gene transfer is us delivering a piece of DNA to the boy's body. I'm not modifying the DNA in the boy's body. I'm giving them a new piece of DNA. So it's almost like a transplant. And just like with transplant of an organ, like a kidney, your body can reject that transplant in certain scenarios. And we've not necessarily seen that with our gene transfer therapies, except for in this case of immune-mediated myositis, and in the cases that have been observed, whereby the body did not identify this new protein as belonging to itself. And so it was viewed as foreign. And the body said, this is not my protein, and I'm going to react against it. And so when that occurred, it led to irritation and inflammation within the muscle that ultimately required treatment. And so the goal is obviously to set our patients up for success and prevent them from experiencing that sort of an adverse effect, in hopes that that new protein that their body is making from this gene transfer therapy is effective and accepted.
Thank you. So, and while we're on the topic, in those circumstances where people with that mutation have had immune-mediated myositis, was the damage permanent? Or was this treated and were they safe and treated? And then was there any follow-on damage from that?
Those are great questions. So in any clinical trial program, there is a process by which we as investigators can report adverse effects that are observed, and then we continue to communicate with the company and with the research team and with the investigators about how those patients are doing. And so we've been able to follow those patients over time and see how they do. They did require supportive care in the hospital. There were treatments that were available to be able to stabilize them and to help them improve. And I know that at least in one case, the child demonstrated an initial benefit after therapy, and then unfortunately, after the immune-mediated myositis, where his body did not accept the new protein, he did not. He went back to being similar to his pre-treatment baseline, as of my knowledge of the last assessment for him.
Thank you. And just to plug, this is why when you're delivering the product for the people that are included in the label, why the families need to stick close and get their blood work done so that you can absolutely follow these children and keep them safe. So thank you for that. And then to you, Siobhan. I think in all of the discussions that Sarepta's had with us, they have promised to really work hard to try to treat every single individual with this diagnosis. Can you shed some light on what you're doing to think about this exclusion of 8 and 9?
Sure. So, obviously we work with safety in mind first. I think that is clear in the way that we approach this. And, we have some work to do to understand if we might be able to dose this population in the future. So right now we're taking advice from experts, advocates, and families about this topic before we move forward with a study. We think that's the wisest course right now before we charge in.
All right, thank you, Siobhan. And Siobhan, in discussion, we've had some questions about what are top-line results? What does that mean? And are you doing further analysis of the entire package? And then when will how long does it take? When will it be available? Can you give us a little light on top line and then what happens after that?
Sure. So top line really reflect the earliest reading of efficacy and safety for a study. After the top line data are released, more analysis of that data occurs to prepare for publication and medical meetings where the data is presented. So more data is going to be released at a medical meeting coming up, and a manuscript is also in development with the goal of publishing in a medical journal next year.
And while we're on the topic, the next question is what does Sarepta need to or required to present? What kind of data? Is it top line or is it deeper analysis to FDA? And then what is the timeline for FDA? I know we can't really have date certain, but what is the process for FDA for submission and then the FDA response to that?
Sure. So I think the FDA has indicated that they're open to a label expansion pending a review of the data. So we've already provided them with top line data while we work quickly to prepare a formal submission. So we will be requesting from the FDA that they review this with the idea of expanding the approved indication such that there's no limitation based on age or ambulatory status. So we believe that this is supported by the data and that the therapy's mechanism of action, the FDA guidance, their prior precedent, both within Duchenne and outside of Duchenne, suggest that this is a potential. So we're going to be seeking that a traditional approval for as broad a label as possible, as quickly as possible.
Thank you. And it's hard, we, as you know, we in this community would like date certain, like on this particular date, but we'll hear something because we are an anxious group for obvious reasons, because we certainly need treatments for all of these young men. So, but we should hear in 2024 and hopefully reasonably early. And I think that's probably the most vague comment I could probably make about when we'll see the FDA. Would you agree with that?
I would agree. I mean, I think we have the top line data now. More work needs to be done on the full data set, and there's a lot of data in here that's really compelling and worth spending time with. Until the FDA completes the review, we can't really speculate on the timeline, but we can say we're moving with urgency to submit, and that they have indicated this a willingness to move as quickly as possible as well. So, and we're seeking the broad label. we're going to be communicating timelines, Pat. We don't want people to be out there wondering for too long. If we have information to share, we'll share.
No, we appreciate, Siobhan, we appreciate that you've been straightforward in telling us timelines. And we're, as I said, very anxious to know timelines because this is an important decision for us. We appreciate the approval for 4 to 5 year olds, but if you're 6, that certainly has an impact. And if you're 3, that has an impact. And as you might imagine, we're all anxious to learn next steps. So back to you, Crystal, we talked about the study that didn't meet its primary in terms of the North Star in the 52-week study. So in your view, and based on your experience, and I'm not asking you to give us deep data into the EMBARK, how long does it take to see change? And what at 52 weeks as muscle gets healthier, it would be cumulative. So do you say, looking into your crystal ball, that, in farther out, we're going to see stability in that North Star or change in the North Star? And what would be an appropriate time frame for us to think, if we got to wait, which none of us want to, what would we see and how long do you think it would take?
So you are asking me to look into my crystal ball, which it's not perfect. It has a little cloudiness to it, but I will say that I am incredibly optimistic. And the reason I'm optimistic is for several reasons. But to start with, we have to think about the disease process. And this is a slowly progressive neurodegenerative condition. And so it is not a dramatic change in wellness that we see in these boys over 1 year. These are slow changes that, like you said, are cumulative. And so with that in mind, if there is a therapy that is stabilizing muscle in particular, which would be success in a degenerative condition, if we are stabilizing a muscle, then that stabilization may not be as readily visible within a short period of time, but really will distinguish itself over time, is my expectation. And if we actually refer back to some of our previous data points, which it does not want to do, here we go. If we look back at the results and the time to rise here, if we're looking at our overall or our 4 to 5 year old or our 6 to 7 year old group here, and we see that they've either improved or they've stabilized at this period of time in their disease, while their untreated cohort is, in the 6 to 7 year olds at least, showing a longer time to rise, I expect that in an untreated group that would continue, unfortunately, to decline, while this other group, if we can see stability, that delta, that difference between those two groups, I anticipate would be more significant as time goes on. So I am very eager to see where our boys are in another 1 to 3 years. And I so appreciate that Sarepta has committed to pursuing long-term monitoring and follow-up of our boys who are treated in both the clinical trial program and commercially. And so I think that we have a lot of good data yet to come.
Good. So, and Crystal, while you're looking in your crystal ball, which makes a perfect name for you, one of the questions that has come in is, do you think it would work better younger, in the 4 to 5 versus the 6 to 7 or the 8 to 9? Can you tell us a little bit about what you're thinking about that? And then a second sort of sub question to that is, one question came in: is this for life? Would this preserve in a certain subpopulation ambulation for the lifespan? So that's quite a crystal ball question, but we'd love to have just your sort of perspective on this.
These are questions that I've had from families. I had from families actually when we were first starting the clinical trials. I had a very, very just eager family who had great questions and said to me, okay, my child is 4. Should we enroll in this clinical trial or should I wait longer? He's doing well now. And so should I wait a little longer until he's older, because maybe his muscle doesn't need it as much right now. So that was a thought that perhaps families now wouldn't even consider, but that was actually quite a common question that I got at the start of some of these clinical trials. And now some of that question framing has shifted to more of what you're asking, which is earlier better? And I don't know that earlier is better. I am optimistic that earlier is better, just like we see in other disease states. What we know in Duchenne is that when a muscle has degraded to the point where it is replaced by fat and fibrosis, which is scar tissue essentially, in this moment in science, I cannot reverse that scar tissue that has taken over in the muscle. And so perhaps if I can preserve this as early as possible, then that would help to stabilize the muscle long term. And so I appreciate that rationale. And I think there's a lot of great work in the community to shift recognition and diagnosis to younger so that we can pursue treatments just like delandistrogene moxeparvovec. That does not mean that I don't think that there is profound potential for benefit in our older and non-ambulatory boys. And I am so grateful that we have work that is being done in that space, because those boys deserve treatment too, and have certainly quality of life and strength that is worth maintaining. And so perhaps younger will be better for longer term distinction from natural history, but I anticipate efficacy in our older and non-ambulatory boys as well.
Yeah, thank you. I totally agree with that. I think preserving quality of life, and I've said this publicly and I'll say it again, I don't think there is anyone who doesn't go to bed at night praying to whoever they pray to stabilize disease progression. Just stop it, right? Just stop it here and don't let it go any further. And I would guess that is at every age and every stage of this disease. So I agree with you. I do think with newborn screening, which we are hopeful to see, and we certainly on the US side are seeing in various states, Ohio being one of them. And since I'm from Ohio, I can mention that. I think we're going to have this question about when do you treat? When is the best time to treat? And I know, and I'm going to pitch this to Siobhan, I know that Roche is beginning the study of what we sort of, in a very friendly way, call the littles, and that's 2 to 4 year olds. Can you say anything about that study, recognizing we're talking about EMBARK today, but are you able to say it's in progress, you have data, or just general comments about the study?
Sure. So the Roche-sponsored study is called ENVOL, and it has only just initiated. And it's actually designed to evaluate people below the age of 4, but it doesn't stop at age 2. It goes lower.
Okay.
It's enrolling outside of the United States, and no safety data is available yet from that study. However, kind of thinking in the context of what we're talking about today and the plans moving forward, we do have the ENDEAVOR study in which we've explored cohorts that are younger than 4 as well. So we'll be submitting all of the safety data in the package that we provide for the FDA.
So theoretically, if the FDA accepts that, the traditional approval could be from diagnosis, even if that's at birth, to an upper end of in terms of age of Duchenne. Is that a fair statement?
That's for them to decide. What we can do is supply the rationale and the support for their decision making, and we do have, fortunately, a constellation of studies that cover a broad range of ages.
Right, that's what I'm saying. You're providing data across from very early little people to adults with Duchenne. And so while we're talking about that, Siobhan, a question for you is about that non-ambulatory study. I think it's called Envision. And in the United States, if I recall correctly, you were recruiting about 20 individuals to the Envision study. Question about, has that been fully enrolled? And do you have any safety data, even limited in that population of young men, to include in your package to the FDA?
Yeah, so to date, we have more than 200 people have been dosed with delandistrogene moxeparvovec in just the clinical trials. We're not talking about those who have been prescribed, and dozens of them are individuals that are non-ambulatory. So the Envision study, which you mentioned is the non-ambulatory and older ambulatory study, is fully enrolled in the United States. It's global. It's a global study. So the rest of the recruitment will be taking place outside the United States. And remember that that's a 72-week placebo-controlled study. The data that we have amassed from the US patients, along with other data from ENDEAVOR, that safety information will be made available to the FDA in our submission.
Great, thank you. And to date, as far as we know, in the study limited and the results aren't out, we haven't had safety issues to your knowledge going forward.
Mine, we should probably direct that to Dr. Proud, but so I can't speak to anything in a placebo-controlled study at this point.
Right. All right.
Yeah, so
sorry, go on.
I can just comment at least on my observations. You're right. It has been recent that that dosing has been initiated within that 303 clinical trial space. And so we're still collecting data, we're still monitoring labs. But from my experience in dosing older and non-ambulatory boys, both in the 103 and 303 clinical trial program, I personally have not seen a distinction from what has been shared and what has been collected and what's in the prescribing information at present.
Great. And Crystal, you mentioned the 103 study. And again, I recognize this is about EMBARK, but in that study, there were adult patients. Is that correct? And they were treated during that period, during that study itself.
That is correct.
Right? Okay. And no safety signals from that study either.
No, and those have been shared as part of that collective data package that Siobhan was referencing as well.
Great, thank you. And Crystal, pull up a slide about the North Star. There's some questions around the North Star and its variability. One question is, as we all know, there's a lot of factors to these little ones when they are doing the North Star assessment. And the variability could be the wrong shoes, the wrong breakfast, the wrong lunch, the wrong person coming with them. Can you just go through the variability in the North Star again and why there is that variability and what you've seen in testing these little guys 4 and 5 years old and their willingness to do this and how it works? Do you do it once? Do you do it twice? And how is this done and assessed, if you don't mind?
So how it's done in a clinical trial may be very different than how it's done in clinic. If you attend a clinic that does the North Star with every clinic visit like mine, and interestingly, my primary physical therapist performs this test both in clinic and in clinical trials. And she would tell you herself that she is much more strict and rigorous in the clinical trial program because she's following a very, very standardized way of providing scores. And so in order to go from a 1 to a 2 is a profound change. And so you have to essentially go from no accommodative technique to a completely typical way to perform a maneuver and a movement. And if you can think about our boys with Duchenne, perhaps they can, for example, lift their head. But if they can lift their head by turning it to one side and then rotating it up, that actually gives them a 1. And so maybe they've gone from a 0 to a 1, but to go from a 1 to a 2 would mean that they perfectly lift their head. And that is perhaps much more difficult than going the other direction. So there's a lot of flexibility in that 1 score. And this has been a great test for concrete reasons to be able to say, you know what, there's not a lot of flexibility here, 0, 1, or 2. But what we've learned through this process, and not just in this clinical trial, but in other clinical trials, is that the speed and the quality within that range of a 1 is incredibly variable. And so some of these subtests, some of these key secondary items are perhaps going to be more insightful and really reflective of what we're seeing clinically. And these are the tests that guide our conversations, right? I'm looking at time to rise from the floor, and when I see that gets to be more than 5 seconds, I need to share with a family that we are in a functional decline phase and we need to brace ourselves for that. When I see a 10-meter walk time of greater than 10 seconds, you know, that is indicative that we are approaching a non-ambulatory phase in the future, in the near future. And so these are tests that guide us just like the North Star would. So each of these tests has their roles.
Yeah, thank you for that. And it is very hard. And I think the predictive power of time to stand in 10-meter walk run is amazing, but also heartbreaking for a family. I can imagine if it was me, I might be doing that every single day to predict. And I think you wouldn't recommend that parents do these tests every day, because it is, first of all, should be done in a clinical setting. Second of all, we shouldn't be predicting ourselves, and leave it up to you, a discussion with you. So I thank you for that. But it's hard as parents, as you might know. So back to you, Siobhan, a couple questions about what is the totality of evidence? What does that concept mean for you as a company and for the FDA? What are we looking at when we say those terms?
So totality of evidence means that all of the data is evaluated, not just the primary endpoint. It includes the primary, secondary, exploratory endpoints, and safety, sometimes other data as well. It's interesting to think about the process that's involved in picking the endpoints, right? And someone somewhere has to kind of put an order to these endpoints. And the primary endpoint is chosen before the study starts. So all of the endpoints that Dr. Proud just talked through are tools for understanding the impact of a therapy. And you have to make an educated guess at the start of the study which endpoint you want to call the primary endpoint versus secondary or the exploratory. And you might not know at the time that you're designing a study how these and how sensitive the endpoints are, or how they relate to one another or to the disease. You can go on what's been done before you and make some educated guesses. But no matter what, if a primary endpoint doesn't pick up on decline in the untreated patients, it's very hard in neuromuscular disease to show a treatment benefit on that, based on that endpoint alone. So totality of evidence allows regulators and those that are evaluating data to look beyond just the primary endpoint and look at the fuller picture, the full data set across all studies, all endpoints. And it's important to know that regulators have that ability to evaluate from the standpoint of totality of evidence as they evaluate the risk and benefit of a therapy, and they can exercise their judgment in determining what's needed to meet that standard for approval for therapies in diseases like Duchenne, where there's so much unmet need.
Yeah, thank you for that, Siobhan. I think that during the AdCom, the previous AdCom, or the AdCom prior to the approval of ELEVIDYS, I think it demonstrated, at least, and you can come and agree or not, that it demonstrated from the Center for Biologics, Peter Marks and his team, a willingness to understand that this is a slowly progressing disease, and that we really need to look holistically at these young individuals to see and to answer the question, has this provided benefit? And I think that during the AdCom, those of us who were able to watch that AdCom and participate in it, some of the videos demonstrated to us things that we wouldn't have, first of all, as parents, been predicted to see in our boys with Duchenne, but also that's real world evidence. And I know that totality of data you're talking about is the data from the study, but also I think this is where I think advocacy is very useful for patients and families to tell their stories about what they see. So in all of that, and they certainly did tell their stories on the advisory committee meeting, do you foresee that not meeting that primary endpoint could be detrimental to the FDA's decision making in light of the totality of data? And B, is it a precedent for the FDA to hold another AdCom, or would that be a possibility for them to hold another advisory committee meeting on their next decision?
Yeah, so I think I'll answer your last question first. People are already asking us about AdCom, and we haven't even submitted yet. So right now our focus is on actually submitting the data and efficacy supplement. And we believe that the data are straightforward and that another advisory committee will be unnecessary. That's our opinion. And we're excited to make this submission. I think it's been years in the making to try to serve the needs of this as many people in this community as possible. And fortunately, the FDA has a history of taking totality of evidence approach, especially for rare neuromuscular diseases, devastating diseases with high unmet need. They're enabled to look across all of the endpoints, primary, secondary, safety, et cetera, all of the studies, and not having to look at a study with the primary endpoint in isolation. So we in some analysis, we found that there are more than 20 products approved between 2018 and 2021, where the primary endpoint was not met and approval was given. So there is definitely precedent there, which is encouraging.
Yes, thank you, Siobhan. I too looked at the data of approvals, both on the CBER and the Center for Drug side. And indeed, people who've missed their primary, but the totality of the evidence points to benefit, they have made those decisions. So we're very hopeful that when they look at the totality of evidence here, that they will understand the degree of benefit that we see in the data that we have, and certainly looking forward to additional data in people being treated of all ages and stages. I think that's really critical going forward. There is a question, as you might imagine, this is for you, Siobhan, about if there's an FDA approval, or I should say, when there's an FDA expansion of the label, does that reach into Canada, or how do you when are we going to see outside the US possibilities of receiving this therapy?
Yeah, great question. Roche is our worldwide partner in the effort to bring delandistrogene moxeparvovec, or Elevidys, forward. And they're working with health and reimbursement authorities to explore next steps for the potential of access in different countries across the world. We're responsible for United States. So I think it would be best for people that are, have called in or who are listening from other countries to reach out to their local Roche affiliate to kind of talk to them and see what their plans are for regulatory engagement. And so I can't speak to Canada specifically, but I know they could probably address that question.
Yeah, I do know that Health Canada is passing laws about rare diseases and trying to invest in them. So, but I don't know anything about their regulatory process and how patients and families could access a therapy. Nor do I know if they how they approve once a drug is approved in the US, what that means to their regulatory agencies. But I think all of that hopefully is, again, a possibility that advocacy should at least ask those questions of Health Canada and other countries so that they are able to access the therapy. Crystal, just I know that and this is a different study, but in Envision they're using performance of the upper limb. Can you, while we're on the phone, or on the call, just talk about the performance of the upper limb? What are you looking for there in a non-ambulatory population, so we can assess as a community as this label is expanded, and when this label is expanded, which we're all hopeful for older patients, what is the pool? How is it evaluated? And can you just shed a little light on that?
Absolutely. And I think that, it's important for us to set expectations that might be appropriate based on where the boy is in his journey with this diagnosis. And so I have conversations with the families and with the patients themselves about their expectations and perhaps my expectations and what I'd like to see with efficacy. And that conversation is going to be very different for a 4-year-old, a 13-year-old, a 19-year-old, a 25-year-old. And so in those older and non-ambulatory patients, we think about what's meaningful from a medical perspective and what's meaningful from a quality of life perspective. From an optimizing functional independence perspective, hand and arm function is absolutely critical. Propelling a power wheelchair, feeding oneself, dressing oneself, bathing oneself, toileting, those are activities of daily living facilitated by the upper limb. And so this upper limb module, this pole that's being used, is assessing the strength of the arms and the hands. And we'll have to see, just like the North Star, I anticipate that also may have its challenges when it comes to the quality of a movement, because it's going to be concrete in its scoring system. The other thing that we have to think about, I'll expand your question just a little bit, beyond that poll, is what else is meaningful for these boys, right? So I personally am enthusiastic about looking at pulmonary function testing, cardiac outcomes. Those are critical to well-being and longevity and life. And so I am optimistic that we will see some impacts there as well. But once again, it's going to take some time for us to be able to see that data, because these are slowly progressive changes.
Yes, thank you. I so appreciate that. And Crystal, if you'll pull up your safety, I think it's slide 21, although I was trying to count on the way. So it's the one where you talk about safety.
Mm-hmm.
I thought it was 21, but I may be misrepresenting here. Yeah, okay. So one of the questions around this is no complement, there's the little bullet here where you have no complement mediated adverse events. Could you just describe complement mediated adverse events? What does that mean?
Complement is an interesting portion of the immune system, whereby when the immune system becomes inflamed, the complement can be consumed. And so this is a portion of the immune system that, if consumed, if used up, can lead to problems, inflammatory problems, and that inflammation can occur throughout the body. The way that we've seen it manifested in other gene therapy programs is through things like a drop in platelets, inflammation of blood vessel walls, and so what's called thrombotic microangiopathy is a very long word for a complicated cascade of immune problems that can happen as a result of somebody getting gene transfer therapy. And I think that this is probably vector related. And so what we're seeing, at least in this AAV RH74 vector that's used for this particular product, is we're not seeing that that complement mediated adverse effect.
But thank you for that. And then one question has come in because we are able now to look at the various changes in the gene. As you know, there are many mutations across this very large gene, and some mutations are predicted to have a slower progression. So, Crystal, in your practice, with an expanded label, if you had a child that you knew based on the genetics of this child that would have a slower progression, would you still recommend this intervention, gene therapy?
Speaking from a personal perspective, and if I was in a clinical room with this patient having the conversation, absolutely. This is an opportunity to preserve that muscle function moving forward. And even in the event of a slower progression, there's progression. And for a family that desires the capacity to stabilize and to reduce that progression, this would be an opportunity to pursue that. And I would be supportive of that.
I can't say that I disagree with you at all. I think that that's going to wrap up most of our questions. One of them came in about pricing of the therapy, and I don't think either you or I or anyone on this call could answer that question. So there's one last question we'll take on, and it is: my son is 7 and still ambulatory. I believe he's already declining. Keeping in line with the conversation about stopping progression, if this were to become available for his age, do you suppose he would remain ambulatory for a long time? And I think this gets into the durability, which Crystal, you might want to comment on, that I don't think we know the long-term durability. These are all things that we'll learn along the way, but please comment.
Absolutely. So you're right. We don't have 10-year data yet, but we do have many years. We have at least 4 years of data from the first 4 boys that were dosed at Nationwide Children's. And we've been able to see that durability at least over that time period. And so I'm optimistic that we'll see that durability going forward. And you're right. I don't know that I can say how long that boy would walk for, whether there would be a slower decline or if we would see them stay ambulatory. But what I can say is this: right now, I have a very clear understanding of what natural history is. And I love the fact that after treatment with delandistrogene moxeparvovec, I can say to a family that I don't know the answer to that question. And this is one of those scenarios where I like not knowing, because it means there's optimism and there's hope for the future.
Yes, and hope, hopefully, as we go along the way, that we can do combination therapies, because as you know, there's antisense oligonucleotides that are approved. There's other therapies coming along, and I think together we'll work forward to combination therapies. And one day we will be able to predict, and hopefully predict for ambulation for a very long time, if not a lifetime. I can't thank you enough, both of you. We've hit our hour mark. Siobhan, thank you so much for coming online and talking to us. And Crystal, we so appreciate all you do for the families that you take care of. You might have a line at the door this afternoon, but thank you so much for your kindness kindness, for your time, for taking time out, and for being so willing to talk to this community about what you see, what you're learning, and always being forthright about timelines and hope. So we are very hopeful for the FDA's decision in our best interest soon enough. And if there's anything we as a community can do to be supportive, we're here to help. So thank you very much. And thank all of you for joining us. These webinars are so important to all of us in this community as we see the hope that we have on this horizon. Thank you again for joining us.
Thank you.
Thank you. Take care.